Seeing God in biology. I'm not going to go to specific revelation tonight, the scriptures, like I usually do. I'm going to natural revelation and what the biology tells us about the knowledge of God and the fact that there is a God. If there's one thing that could be said about our age is if we were to, I don't typically see statues of Zeus or Apollos around, but if there is a God of this age, it would be science. And if there's a gospel of this age, it would be evolution. You know, and you kind of read it, like when you read science and things like that, one of the things that tickles me is that if they find water someplace, they get really excited. Have you ever noticed that? Yes. And if they find organic molecules on a comet, they get really excited. And, you know, can you imagine what it would be like if they found water and, you know, organic compounds all at the same place, how excited they would be? This egg I cooked, just in case I dropped it, it wouldn't have messed. But you need to imagine what's in this egg. Water and amino acids. Do you realize, I think this way, but I was thinking one time, do you realize everything you need to make a chicken is in here? Right? I mean, if this is fertilized and becomes a chick, everything that's needed to make the chicken is in this egg. You might even say that if you ordered a chicken from IKEA, this is what you would get. All right? Now, here's the thing. If it is fertilized, it's like getting the kit with its instructions, right? Now, I mean, from a scientific viewpoint, if I were to sit this egg here and let's just wait for something to happen, right? It's going to get rotten, right? What is it about a fertilized egg that makes all the difference? It's more than instructions. It is a whole process that knows how to take the amino acids and the calcium and the chemicals and things like this in this egg and make a chicken out of it. And this is the path we're going on tonight. Not the whole making the chicken thing, but that one of the key things in biology that puts a lie to an atheistic worldview is that biology runs on information. Not physics, not chemistry, information. So we're going to begin here. What's this? Anybody? SOS. I don't see SOS up there. But you're right, it's SOS. It's code, right? And not only that, but it's two codes, right? There's the Morse code. There's the ... dash, ... or DDD, DDD, depending on how you want to do it. But it's also SOS. And SOS is a code for international distress. Okay? Now, here's the thing. Is there anything in the universe that mandates that DDD is S? Is that mandated? No. Samuel F.B. Morse said DDD is an S, is an O. So let's ponder the telegraph and Morse code. Over here, let's see, how's this going to work on you guys? Okay, so over on this side is the sender. He is sending a message. And then it has a power, it's going over through power lines, and over here, there's a guy who's very happy in the message that he's receiving. Okay? Now, both sides have to have the same code book. Right? For this to work, you have to have the sending station, you have to have the receiving station, you have to have a code, but both sides have to have the same code book. I don't know if any of you are decoding you, but up at the above, what he is, the message he's sending in Morse code is, Jesus loves you. All right? Okay? So, now the same must be Morse. Like I said, a dot or a D is a short tap of the telegraph key. A dash is a longer press of a telegraph key. He decided that a dot would represent, a single D would represent an E. Why? Because E is the most common letter in the English language. So, not only in his design, he designed it to have an element of efficiency. And therefore, the most common letters that you're going to send over a telegraph have the shortest number of strokes. And it gets on to the ones that are very, very long because you don't use them so much. So there's some design, but again, there is nothing in the universe that mandates this code. It was invented, and it was invented by a mind. Get that, understand that. Codes are invented by mind. So, for a working telegraph, you need a sending device, right? A little clicker. You know right? You need a receiving device, a little tapper that goes like this. When the guy hits this goes up like that kind of stuff. You need the wires. You need a means of transmitting this click to this tap. You need power. And you need the same code book on both sides. You take any one of these away, and it does not work. So the whole system is designed. Not just the code, but the whole system is the product of information. And you're saying, well, that's duh. All right? Well, the thing is, I'm going to show you some videos tonight. And the people who produce the videos don't see the obvious thing that I'm telling you right now. They see it as, hey, if we get water and amino acids together, we're going to get life. I mean, life is just a matter of just add water, it almost seems. So, you need a designer to create a code and make sure the equipment can translate it and that each side uses it. I'm repeating myself. But this is really, really important. Code represents information. Information comes from intelligence. I am not aware of any information that has ever not come from intelligence. So, all right, codes are arbitrary. Right? I'm repeating myself, but again, understand this. So what does this say? Anybody? You were really good on the Morse code. What does this say? Well, the information is there, and if you know the code, you can retrieve the information. And for your information, I designed this code. Right? All right. So it's a very simple substitution cipher. You take each letter and you back it up by 1. So the K becomes a J, the F becomes an E, and here we are at Jesus loves you again. Right? So, all right, but language also is code where sounds communicate meaning. I mean, every time, when you were around intelligence, it's all code. Jesus, Yesu, Yeshua, Hebrew, Yeshua. Right? Our letters code for sounds, they code for meaning. It's all code. When I say door, that's a code for that thing over there that I'm going to walk out of sometime tonight. But door, it's code. Code, code, code. Information, intelligence. So, codes are the product of intelligence. Code transmission is the product of intelligence. Sending and receiving require the same codes. Because if I say door and you speak Taiwanese, you don't know what I'm saying. And if you know something as a code, you can decode it. And codes are designed and require a designer. So I'm going to show you 4 videos tonight. These videos have all been produced by secular organizations. The first 3, in fact, have been produced by Harvard University. And they are going to show a cellular equivalent of a telegraph. There is going to be a sending code. There is going to be something that takes that sending code and transcribes it into something else. And that something else is going to go to another place where that code is going to be read and is going to create a protein. Code, 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 with machines in between to make it all work. So I'm going to start with DNA transcription. You've heard that DNA is your genes, right? Well, your genes are nothing more than proteins. You know, DNA is a code for proteins. DNA is responsible for your hair, your fingernails, all the other proteinoid things that are in your cells and so forth. That comes from DNA. DNA contains lengths of nucleotide codons that code for an amino acid. Then we have messenger RNA. It is a chemical string that is made from DNA that is a copy of a section of DNA that's sent off outside the nucleus. Okay? Then we have translation, where that messenger RNA is brought into a ribosome, and it starts bringing things in that says, oh, does this match? Does this match? Oh, this matches. Let's ratchet it over and add an amino acid to the protein. These are amazing videos. Okay. And then at the end, I have this thing called the inner life of the cell, which is just has pretty music and just kind of a walk through a cell with all kinds of things going on. So, the players. DNA, a sequence of chemical codes that happen to be labeled G, A, T, and C. And if the code is TTT, that codes for an amino acid called phenylalanine. There's going to be a test on this at the end. Sorry. You don't need to know this, okay? But some of you like this stuff. I like this stuff, so I'm throwing it in for you. But what's cool is there's a sequence in DNA. By the way, DNA strand is 5 feet long. One DNA strand is 5 feet long. It's 5 feet of information coded on there. And as you walk along there, there is a codon called 8 that is a sequence of A, T, and G. And that says, start here. Okay, then there's a bunch of codons, and then there's a TAA which says, stop here. And between the ATG and the TAA is the code for the protein. By the way, we are told that this is all by accident. Right? Honestly, we are told this is all by accident. The messenger RNA carries the code, and the transfer RNA they're these curious little triangular molecules that have bumps at the bottom. And the bump is like a key. And you know, that bump, if it corresponds to the messenger RNA, a couple of bumps in the messenger RNA, it's a match and it contributes the amino acids. So let's watch the first one.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
So that's the start of the telegraph, right? We got the code's been copied to messenger RNA, and now let's make a protein from that.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
Isn't that amazing? I mean, do you realize that as your hair is growing, that's what's going on inside your hair follicles? Is DNA is being used to encode the various proteins for your hair. It's being sent out of the nucleus. It then goes into a ribosome, and the ribosome produces the protein, and your hair starts growing. Okay? This is code, right? I mean, just if we're talking about a code in the DNA, and then we have 22 different green molecules, one for each amino acid. And it's got to match up to the messenger RNA, or you don't get the protein that the DNA said that you should get. And to make this even curiouser, right? Those transfer RNAs, they're made out of protein. The ribosome, the component parts are made out of protein. And guess where those proteins are coming from? DNA. You know, and not only that, I mean, the thing that, I ponder this, and I go into worship. I mean, I don't know if you can appreciate this. This is a worship experience for me. But like there is in a bacterial cell, there's a flagellum. Okay, and the flagellum has a motor and it's got all this stuff in it. But the thing is like it's constructed from protein. But think about building a house. You start a house. What happens if the shingles show up first? You've got no place for them. They take up space. It's not efficient. I mean, among all this other stuff going in the cell has to know what protein is needed where now. And how that happens is still a mystery. But it happens. That's why you get a chicken. That's how, you start with a single fertilized cell and suddenly, it divides and divides and divides, and all of a sudden it says, oh, hey, I can't be what I've been. I need to become a feather. I need to become a heart. I need to become part of the lungs. And there's all this differentiation that is happening within the cell, and it's all information. Okay. All right. I don't know how, this next one I'm going to show you, I don't know how a scientist can look at this and say that it all just happened, right? DNA replicates, you know that, right? Okay, I mean, that's how when cells divide, you have to make copies of all the DNA. And I used to think about, well, there must be some interesting process that allows for the DNA to separate and rejoin and matches itself and so forth. What do you see? What goes on? This is how DNA is replicated. And by the way, all the parts that are here are made from protein and have to be constructed by DNA also.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
Spins at 10,000 RPM. I mean, it's like a jet engine. Okay, one strand has to be copied backwards. Okay, I mean, you talk about chicken and egg, right? You need this conglomeration, this machine, to get a copy of DNA, and DNA is there to be able to construct the machine that does the copying. And let me tell you some of the other things that are going on. Think about a shoebox, and you put 42 5-foot long strings in there. Shake it up. Okay, you start pulling them out. Have you ever heard of knots? Right? There are systems within the cell that before the DNA is replicated, take out all the knots and bind it up into what are known as the chromosomes. That's just a whole deknotting process that has to happen before the cell divides. When that thing is spinning, if you think in terms of if you have a twisted, couple of strings twisted together, and you start pulling it, right? Know how it knots up? There are things during replication that detect those knots, and they come along and they do little snips to relieve the pressure and reassemble the thing. I mean, we are talking about information and control and procedures and things like that, the likes of which are beyond comprehension. And like I said, I look at this and I go into worship, because the wisdom that's here, the genius that's here, and just, part of my worship, I think, I look at this and then I think about Jesus turning the water into wine. I mean, the same genius that made this is the genius that says, oh, I know how to turn water into wine. I know how to manufacture chemicals and things like that and put the mix in there so that it looks like it, feels like it came from grapes and it's the finest vintage that has ever been ever. Because Jesus is the wisdom of the Creator that knows this. Now this one is just pure joy. I mean, this is without explanation, except that I will tell you this. There's a code involved here too. This is code where you have this white blood cell that's floating around in your blood vessel. And suddenly there's an infection that's calling it. And there is a signal that is given to the white blood cell to cause it to collapse and go to where it needs to go. And this is sort of like a picture of all the different things that are happening. The signal arriving and the proteins being made and transferred and things like that. And it's also some pretty nice music. They're constructed to, when a protein needs to get from one place to another, it constructs a road. And you got that little walker guy who's carrying a sack full of proteins to the place where it needs to go. How do it know? You know, this is the mysteries inside the cell, and I trust that having seen this, your faith that there is a God and that science is a bunch of hooey when it says there is no creator. I mean, science has its place. I mean, science is what's discovered this. But let me tell you, this is information. And this just tells me that there is a creator and that he is a genius. So, you need a sending device. Well, we got DNA scanning. You need a receiving device, that's a ribosome. You need the means to transmit and sending action, that's messenger RNA, just like the telegraph, right? You need power, and that happens in the cell through this thing called adenosine triphosphate, but you need the same code book, or it does not work. So, again, these videos were made by secular organizations, and they are a significant challenge to the materialist worldview that these things just happened. But these point to a creating intelligent, capable of designing the living cell. And the desire required thousands of proteins to broker the function of the cell, created the codes for those proteins, and created a mechanism that used those codes to make the proteins to make us. And evolution, right? A change to an organism requires a code change. I mean, I'm a computer programmer. I do software for a living, okay? And a new release requires a code change. It looks like it's evolution, right? It looks like it's evolution because there's incremental change. But each incremental change required me as a designer to acquire information and to encode that information into a code change. So evolution, if it exists at all, requires a designer to take things to the next level. You know, and quite frankly, I think what happens, I mean, if you want my view of what happened on the earth, the Lord created the cells. You know, and then periodically comes along and puts in the new versions, you know, and so forth. But anyway, here's a challenge for you. If you are reading some news article or something like that about evolution and so forth, where it reads evolution or where it reads nature, substitute in God. And it works. It works better. Because what the evolution is, what this whole thing they fail to understand is they talk as if evolution is guided, that evolution has a goal. But the actual truth of evolution as they define it is random variations that have no direction whatsoever. And you don't get information out of that. So anyway, thank you. I trust this has been interesting to you, and that it increases your faith. Thank you.